So what I want to talk today is something that's has been a long quest for in the field of delivery, and that has been for getting into selective cells and deliver different cargoes. And one of the main cargo that my lab is interested in is of delivery of messenger RNA. So I do have a conflict of interest. I'm a co-founder of 2 companies and also an SAB member for another company. Like I said, I think I'm so grateful to speak today in this Pieter Cullis Invitational lecture. Pieter is a legend in the field, has basically initiated this field that is helping us around and next generation around to pursue something very exciting. And we are so grateful for all he has done in terms of scientific innovation, but also mentorship for so many students who have gone to industry, academia, and for people like me who didn't come from his lab, but he has supported as you know in the development of my own career. If you look at Pieter's work, this was pre-70s, but Google citation is just from the 1990s, and you can see that it's growing and growing and growing. We now have, and he has an H index of 135 and a huge citation index. So I'm so grateful to be invited to give this lecture. Not only does Pieter and I have a common sort of love for lipid nanoparticles and these delivery modalities, but we also share the same birthday. So I never forget to basically call on Pieter on 12th May because I remember that we share the same birthday. And not only that, I mean, the same birthday is not just shared by me and Pieter. We also have the British Prime Minister who also has the same birthday. So as you can see, we are, we share a common love for lipid nanoparticles, but we share a birthday with the British Prime Minister as well. And recently I found out that I end up sharing the year with him as well. So when my wife looked at it and she was telling me that she missed out marrying the British Prime Minister, I thought I'll tell her that I missed out marrying a daughter of a billionaire, which the British Prime Minister is married to again. So anyway, this is a common sort of like a little trivia that I wanted to share with you. But as I said, I'm so grateful for all that Pieter has done. And if you look at his work, I was going through some of his papers, and this is as early as, I think, 1974 or 75. I know Terry Ellen is here. She has done a lot of work with Pieter in this area. And nowadays, like a lot of companies on Wall Street, they come to us and say, oh, have you heard about these new fusogenic sort of lipid? They even have, I think, trademarked some of this fusogenic language. And it's funny because Pieter in 1978 published a landmark paper on how lipids have a fusogenic capability. And that sort of is a hallmark that when ionizable lipids were found, they had an ability to escape the endocytic compartments. And if you look back, I feel like even, we published recently a paper on structural polymorphism of lipid nanoparticles, and lo and behold, Pieter has already worked on that area in 1985. So to speak that there has been already existing a lot of work that has been done, even before some of us were born, which is elegant and still holds true today and has laid the foundation of future sort of work that is going on in the field as well. Even delivery of DNA, I think that's getting a lot of traction nowadays. And then reaching on patro, and as we know now that we have all these vaccines out based on some of the seminal work. Even corona formation seems to get a lot of traction. And as you can see here, that there was a 1998 paper on how liposomes can interact with blood proteins. So these foundations have our field is set on strong foundations, and that's why I feel like it keeps on growing and building on it. And I think as we move further, the dream that we drug delivery scientists have to deliver a cargo to each spot in the body, which per se has been the rate limiting step. We have a lot of cargoes today, but there are only few ways we can deliver it and to few organs. And you could see even in the biotech sector that the technology, therefore, is limited to those organs as well. The kingpin for the LNP space has been the ionizable lipid. Again, a lot of work has been done through academia, through also, I would say, biotech companies who took a lot of risk to basically identify some structural properties of ionizable lipids. And I still remember when I started in the field, there were lipids, but they were not potent enough, or we didn't know what their structure-activity relationship was. But slowly we have started to come across certain relationships. For example, a requirement of a head group, which is some sort of an amine that can help in the endosomal escape, but also an interaction with an RNA, a linker, and how long those linkers are, if those linkers can be biodegradable. And then the tails also are really important in where, not just where the delivery is happening, but also where the biodistribution of the lipid is happening as well. And with these lipids that interact with the RNA, we do have other lipids that are helping. So these are the PEG lipids that give the stability to the particle and also prevent some of the opsonization that happens. Helper lipids and cholesterol, and individual lipids have their own sort of effect on not just biodistribution, but also in terms of cellular tropism. And more work now has been showing that some of these sort of excipient-like materials not just help in the process of endosomal escape, but also might trigger where a particle not just goes in a tissue, but cell selectively might distribute in a very different way. And I think it's just the beginning of a very exciting new technologies that would jump up ahead. And as you know, that how we make these lipids is we put the lipid components in ethanol, the RNA component in an aqueous acidic solution. We mix them through rapid mixing. And again, a company that Pieter started, Precision Nanosystem, has made a lot of headways in making some of these devices. And that has actually helped in scaling up a lot of these lipid nanoparticles. There are also methods like T2 mixing that are being used to really scale these materials up. Otherwise, it used to be very hard to make these LNPs, again, in a very scalable sort of manner. Once you expose these materials to the cell, again, because the lipid now switches back from a neutral pH to acidic, at an acidic environment to a positive charge, it interacts with the endosomal compartment, and that leads to an escape. So the advantages have been many. The modularity of these LNPs, we have seen that play out in real time. They can package large RNA, they can package an siRNA, so we have on Patro, they can package an mRNA, so we have vaccines. And now, there are clinical trials happening through Intellia and Verve and many others where they can package RNA and like a small guide RNA for gene editing. So the modularity of the LNPs is not stuck with just one cargo, but multiple different cargo. And the reality is that these, our studies have been focused on delivery of DNA in the 90s. And we still want to figure out novel ways to deliver DNA as well. So I think there is an interest going back to that, but there are certain challenges, of course, in delivery of DNA. They are biodegradable. You can redose. We have received multiple doses of these vaccines, and scale, it's a different scale now, right? Like just 3 years ago, the scale was that we could just get to maybe like 100,000 people, but now billions of doses can be made. So if we find the next novel lipid that goes to the lung or the heart or the brain, the scalability would be such that we could give it to a rare population with a rare genetic disease for the lifetime very easily. So that sort of has now even expedited some of these technologies. And the focus has been on different cargoes, from self-replicating RNA to SI to CRISPR-Cas. And as more and more complex cargo come in, we do have this incredible delivery system that can at least package and deliver to some organs. But as much as has been done, one thing that still remains a challenge for us is our delivery capacity is limited either by an IV dose, which then the particles go to the liver. And even in that case, we use sometimes non-inflammatory drugs because there is a chance of inflammation after you redose multiple times. There are these vaccines now, which is at a muscle level. So for the next generation, especially for the scientists who will be like grad students and postdocs here, what we now can foresee is, can we deliver to other organs, maybe the lung, the eye, the liver, the heart? I mean, liver is sort of done, but there are some cell types there. And then within those organs and tissues, can we selectively deliver to some cell types? That still is a big challenge. And I think slowly and steadily, we could come up with strategies, and the quest continues to deliver to such organs as well. One way to deliver would be, through a direct sort of localized environment. So I'll give two examples of the eye and the lung, where localized delivery could help us deliver to certain organs, as well as certain cell types, and that can lead to new therapies coming forward as well. If you can in today's world, the challenge is delivery. So if we can get a specific delivery to a cell type, that unleashes, because there are a lot of cargoes waiting and a lot of targets waiting. The challenges can be delivered to those targets. So one way is that, and then I'll talk a little bit about how can we use the newest and greatest technologies to go beyond some of the hepatic targets after an IV administration as well. So the first example I'll give is drug delivery to the back of the eye. The eye is composed of a neuronal retina. Again, it's a beautiful piece of tissue which has neurons and arranged in different layers, and an epithelial cell line called the retinal pigmented epithelia. So the photoreceptors are the light sensing sort of cells. So the light comes in, goes back to the eye, so across these different layers, and the photoreceptors then convert that into an electric signal that goes back to the brain and where the image is processed. Every day when we wake up, the photoreceptors outer segments sort of are lost and are eaten up by the RPE. So these epithelial cells are highly phagocytic in nature. They're also called the liver of the eye because they eat up the outer segments that are, again, every day when you wake up, these are lost and new are regenerated. So if there are mutations in the RPE, then the outer segments start to not get digested and you see a loss of photoreceptors. Or if there are mutations in the photoreceptors, you'll see a loss of these photoreceptor cells. They tend to die. If they die, there is no, conversion of the light, and that leads to blindness. So what my lab has recently focused on, by recently I mean for 6 years, I think in academic research setting, 6 years is recent, it is in the space of retinal diseases. So As you can see here on the right, these are the rod cells in the normal retina. If there is an inherited disease or mutations in them, which led them to these sensitive neurons to die, you see a complete loss of this layer. And this leads to a progressive loss in the vision. So as you can see here, this is a dog, Polo, from my grad student, Marco, who now is a postdoc. He basically showed that how you start to get a tunnel vision once your photoreceptors start to die off. So one way to prevent that is to correct some of these genes, or through gene augmentation or gene editing. And there are a lot of ocular treatments. One treatment for gene therapy that was approved in 2017 was based on an AAV, which was subretinally injected. And in the disease was in the epithelial cells, so the retinal pigmented epithelial cell. And there's a mutation of an RPE65, which was given back, and Luxturna performed really well. But the challenge is that you can have one injection through a subretinal route. The LNPs can carry, large cargo, which an AAV cannot. You can redose with an LNP. But with an LNP, the expression, especially of RNA, would be limited. So the best application, if we are giving subretinally, would be for gene editing applications. And so there are multiple routes of administration into the eye. One route is the subretinal route, in which you form a bleb in the back of the eye and you directly give your dose into the back of the eye. So it's highly invasive, it detaches the retina, but it works. You can form multiple blebs. Luxturna was given through that. And scientists, especially the scientists I collaborate with at the Casey Eye Institute, are basically able to do this very confidently and have been very successful. The other route is the intravitreal route, where you inject it into the vitreous. This, as much as hard this also seems, this is done routinely for like wet AMD, and also recently there is a dry AMD product. All of them are given intravitreally, like an antibody or things like that, and they can be given monthly. But the challenge is with lipid nanoparticles and such, there is a viscous environment here, and the particles get stuck and will not reach to the back of the eye. There are also other routes like suprachoroidal that are also being applied, but they can also have their own challenges. So, this is just showing the viscous environment and where the nanoparticles would be trapped. So if you take a traditional lipid nanoparticle, and take a Cree mRNA in an AI9 mice, which has a TD tomato with the LOXP sites. So if the Cree mRNA is expressed, the expression of the recombinase edits out the FLOXP site, and then you can see the TD tomato. So this is fundus imaging. You can directly visualize in the eye of the mouse. So, it's an in vivo imaging kind of a strategy. Once you cut the section, again, these are the different layers of the eye, and where you see expression is just in the retinal pigmented epithelium. And I'm not surprised, because, these are the liver of the eye. So of course the lipid nanoparticles end up there. If you give an intravitreal injection, the particles do not reach to the back of the eye and are stuck at the outer layer only. So with my grad student, Marco, and collaborator, Rene Reals, we started to figure out, can we bypass some of these methods? So there were two things we wanted to do. One, we wanted to inject the particles through an intravitreal route so that we can do multiple injection. And then second, we want to hit the photoreceptor within the eye. So there are two challenges that we are going after. So what we did was that, since the lipid nanoparticles end up through the subretinal route only in the RPE, we said, what if we took a phage library, which has a diversity of peptides, 10 to the power 9, injected intravitreally, and took out the back of the eye and see if some of the phages bind to the back of the eye. And from there, do the panning process, grow those phages basically in an E. coli, identify those phages, and keep on enriching those phages till we find certain peptides that could actually cross after an intravitreal injection and take the phage back into the back of the eye. This work, again, was published recently in Science Advances. So what we found was that if you inject intravitreally a phage, empty versus the one containing 7 mers, there was a 100-fold difference in ability of those phages to cross by. So what that suggests is that if you have a peptide, certain peptides could cross the vitreous membrane and vitreous of the eye and go to the back of the eye. And through different sequencing and panning processes, we started to understand where the amino acids were, what the amino acids were enriched with. And we identified certain peptides that were highly enriched and had the ability to reach to the back of the eye. We then took those peptides and basically looked at their binding ability in a cell line. So the RPE cell line as well as the cone cell line. And what we found interestingly was that in the RPE, there was no common theme within the peptides. These are the different peptides that we put under computer simulation. But they were, when we were looking at peptides that can reach inside the photoreceptor cell line, there were, they completely superimposed on each other. So there were certain properties within these peptides that could traverse the membrane, and basically they are also binding to the neuronal cells within the eye. So we took some of the best performing peptides, like this peptide 42, which works really well in 661W cell, and we added a TAMRA dye on it. And now we injected to validate our targets. We injected those in the eye. And after an intravitreal injection, we see that within 6 hours, you start to see a very nice peptide localization into the outer nuclear layer where the photoreceptors are. If you just took the TAMRA alone, you don't see any such localization. And if you did a subretinal injection, you start to see even better nuclear localization and outer nuclear distribution of these peptides. So we were very excited about that. Because you can think about that, just these peptides, you can attach certain oligos like an SIN and ASO, and after intravitreal injection, you might see knockdown in the photoreceptor cells. What we did was we took, because our interest is in cell-selective delivery of lipid nanoparticles, we took these peptides, conjugated them on the PEG lipid through 2 different conjugation methods, tested how much ligand density was important, and figured out like around 30% ligand density, we start to see a very good expression in the subretinal space after a subretinal injection. Even intravitreally, we saw some expression that was in the Mueller glia region of the eye. So we took the best performing LNP, and what we have done is we have done further surface modification. So this is some unpublished data which will be published soon, that lipid nanoparticles have their own, if you can modify them in certain ways, you can change their distribution to the photoreceptors cells. So as you can see, again, this is the Cree model. You inject our modified LNPs. This is the RPE section, but the photoreceptors light up after expression. But the traditional lipid nanoparticle will just remain stuck in the RPE layer. We have done further studies where our modified LNPs can express pan-retinally. So this is an expression of the whole retina. You take the retina, you cut it out, and you can see the RPE and the photoreceptors beautifully light up once we modify these lipid nanoparticles. We can then take these lipid nanoparticles that have a peptide on the surface, and I apologize for people on the West Coast, might be lunchtime there, but we find that once you inject LNPs through the subretinal space. This is again the clinician who also does injections in people, injecting in a non-human primate. And as you can see that he forms a bleb, injects, and you can see the exposure of lipid nanoparticles in that bleb. And interestingly, what we find is a beautiful all across this bleb a very wide range of expression in the RPE, but also photoreceptors within the eye. We don't see a lot of damage in the eye. We've, again, in ARVO, we'll be presenting some new data where we have our own novel lipids that can reach to the photoreceptors with and without immunosuppressants. And we see a very high expression in the photoreceptors using immunosuppressants as well. Again, that's the future sort of publications that you'll see in this space from us. So this is just cell type verification. So cone arrestin, we see some expression in the cone. Then rod arrestin is a marker of rods. You see a huge expression in the rod cells of GFP at this point. Some expression in the RPE and a little expression also in the Mueller glia within the eye. So what I'm trying to say is that as we open this new sort of chapter on the ocular space, we think that in the eye space, slowly and steadily, we can get a great expression of these lipid nanoparticles, enabled RNA technologies, and from the liver and the muscle, I think there is movement of the field towards the lung, but the eye could also be a huge target, and where we can have a huge difference in terms of using these enabling technology for treating a lot of different diseases. And what our interest is to deliver genome editors. We have been working on different kind of gene editing, and I'll not go into the editing space, but what we have done recently is we have taken our lipid nanoparticles. We are currently working on methods of putting a prime editor that has an ability not just to edit, but then give back a DNA into the genome. So, you can solve 80 to 90% of a lot of genetic diseases. But the problem with, again, prime editor, it's huge in nature. So how you encapsulate and deliver and the amount and the efficiency of delivery is still a question. But that's something that we are very interested to do in the eye space. Especially with LNPs, which can incorporate all these vectors in one, all these cargoes in one, rather than an AAV where you use a split AAV vector. So what we have done recently is we have taken the traditional CRISPR-Cas9, and what we have found is that you can actually load the Cas9 mRNA with a guide RNA against, again, the TD tomato, and we have seen a really nice expression into the RPE, lower expression to speak in the photoreceptors, but we are still working on attaching our targeting ligands and other methods to improve efficiency of gene editing using non-viral vectors in that space as well. We've also been able to incorporate some of these prime editors. Like I said, they're relatively big in size. And in vitro, we have shown that we can get a really high delivery efficiency, and we are currently working on methods in vivo. Especially the eye is a little harder space because in a mouse, you can just inject 1 to 2 microliters. So we really need a very potent system to see effect. So we are currently working on that. And we are not limited to just photoreceptors. I think slowly we'll be targeting ganglion cells and other cell types, that are associated with glaucoma or some other diseases, and try to go over a lot of these genetic disorders, as well as some other diseases of the eye. And I think the LNPs would be really effective in treating a lot of these disorders. The second area where we have done a lot of work is in the inhalation space. So we also want to deliver the lipid nanoparticles locally again into the lung, especially because I have support from the Cystic Fibrosis Foundation as well as NHLBI, and we were working on delivery of CFTR mRNA into the lung through repeated inhalation. And we even have some data, which I'll show today, where we have some novel technologies where we just use an IV dose and we can hit the lung as well. So the challenge with making an inhalable LNP is that if you put an LNP in a vibrating mesh nebulizer, it breaks apart. So again, this is like pre-nebulization. This is how nice the nanoparticle looks, low polydispersity. If you nebulize it using a vibrating mesh, then it starts to aggregate, and we also see a loss of RNA. So one way a lot of people, in the field, like people like Justin Haynes and others, have used PEG lipids to stabilize the particles. So if you increase the amount of PEG lipid, the size goes down, the aqueous stability increases, and the nanoparticle mobility increases in like a mucosal environment. So, again, the CF, there are two challenges. There's a thick mucus, and then once you cross the thick mucus, you have to reach the epithelial cell line or epithelial cells to deliver your target. So in this case, what happens is that we find that if you have high PEG or the field has found, then you see a much more diffusibility of a nanoparticle than if you have a low PEG in the system. But the problem is that even though after you increase the amount of PEG, the size decreases and you'll have a very low polydispersity and a nice size of a particle, the expression, if you throw it on cells or exposed to cells, this is 1.5 PEG before nebulization. After you start to increase PEG, the expression goes down. So now you have made the particle so stable using PEG that you do not find an improved sort of delivery, because it's stable once it goes inside the cells. So to overcome this, what we've figured out that if we use a cholesterol analog called beta cytosterol, that helps in packaging of a particle in a polyhedral shape, and that even at a high PEG, that may allow ability of expression. And cytosterol, what we have found and others have been able to repeat it, is a very potent endosomal escape agent. So it really helps in the escape process. So if you compare cytosterol to cholesterol in all these cell lines tested, you see, from 30 to 20 to 10-fold differences based on the cell line. So, and again, we have published this in multiple publications where we show that addition of cytosterol changes the structure of the particle. So rather than a spherical LNP, now you have a polyhedral LNP. And using an assay that helps in identifying endosomal escape called the Galectin assay, you can measure a high. We find that with particles that contain cytosterol, there is a high endosomal escape that happens. So again, what the assay is that, if you take galectin-8, it's a cytosolic protein, there's a cell line stably expressing galectin-8 GFP. If you expose a lipid nanoparticle with an RNA, that galectin-8, which is cytosolic, starts to bind to these punctate structures. So it's an indirect way of measuring endosomal escape. Again, the lipids are not labeled here. It's just that when you expose with plain LNPs on these cell lines, the cytosolic protein starts to go in the endocytic vesicle. When we then add cytosterol, you see a huge improvement in escape. You see a very large number of puncta as compared to control or just cholesterol containing LNPs. So again, the endosomal escape is an issue, and with cytosterol we find that there is a huge improvement in the endosomal escape process. So our agenda was that, can we make a particle stable enough with a high amount of PEG that can withstand nebulization, cross mucus, but labile enough that once it's inside the cells, because it contains cytosterol, it will be able to lead to a higher endosomal escape. And this is just showing in a mucosal environment, a particle with the cytosterol, instead of cholesterol containing LNPs, we see a higher movement in the mucosal environment as compared to the one that has low PEG. And once you nebulize the particles, again, we find that you see a higher expression after nebulization with around like 3.5% or even 4.5% PEG, which the particles also contain cytosterol in them. So once you nebulize the particles, we see a very nice, beautiful expression on 5 different lobes and this work was done by David Kim in my lab, who again is a professor now in Korea. We find that the expression is on all different 5 lobes in the mouse. We then again did this Cree recombinase study to figure out the cell types that we can express into the lungs. once you nebulize the particles, we do find that there is expression in the alveolar as well as the epithelial cells, the ciliated epithelia in the lung. Following which we did a repeat dose experiment, and what we find is that after repeat dosage, we can have expression of luciferase, which is sustained, and we can give a large gene like a CFTR mRNA gene in a CFTR knockout animal, and you can see expression there as well. So currently we are again using things like prime editing and other editing methods to make treatments for CF that may be like a one-time cure. And then we are very excited about it. We have also in the CF space shown that CFTR mRNA works. It's functional using the nasal potential differences, as well as we have worked in the space of ENaC, where again, we see that that can also be another target for the treatment of CF. We've also used these lipid nanoparticles to deliver soluble ACE2, which can neutralize the spike protein. So in the virus realm, rather than a vaccine, these soluble ACE2 can capture the floating virus and neutralize it. And we have some these two publications, or one publication in Advanced Sciences that came in 2022. But we also are looking into different nebulization methods. So we are currently, either you change a lot of formulation parameters to get a stable particle, or you change the nebulization method, because currently the nebulizer that is being used is not amenable for for lipid nanoparticles. Like I said, they break down there, so you have to change the formulation. So if you're using, for example, the current SARS-CoV-2 vaccine, you won't be able to use with the current vibrating mesh if you want to make like a nasal vaccine or things like that. So with another company, we've initiated, it's called Rare Air, where we have a new device that the way it sort of nebulizes the particles is in a way that it doesn't change the size. So the size with the vibrating mesh changes with the new device. Before and after remains the same. The polydispersity remains the same. And the size also does not change once you pass through this method. You can do an EM, and as you can see with vibrating mesh, the particles tend to aggregate. With this new device, we do not see any much change. And like I said, we are very excited, and hopefully we'll be publishing a paper out soon describing this device. And I think I'm very excited about that study. We've also shown that we do not, there's no loss of RNA encapsulation. There's no fusion that happens between the nanoparticles, which usually happens if you are using a vibrating mesh nebulizer. So there can be a lot of applications to study a lot of particles and deliver them. And we see that we can maintain the expression with the vibrating mesh at the same level. You don't see any expression with the new device. It's as good as when you put like a lipid nanoparticle solution, which is not nebulized. So like I said, there's 100-fold improvement compared to the vibrating mesh system, and we are very excited to push that forward. And in the future, I'll talk about that device and how it works as well. So from this, I also want to describe that we've, currently the work we did was using, lipids that have been pre-synthesized by, others. So there was a high need that, we start to have an in-house lipid, and we start to study its trafficking. But also to understand the trafficking pathways, we needed to make a lipid that is much more rational in design. So hopefully soon I'll be describing its structure as well. But for now, we have a novel set of lipid using a new chemistry where we have a proprietary core, and you can change these different R1, R2, R3s for an ionizable lipid, or for a lipid moiety, or for an amine or an endosomal escape agent. So the head-tail linker can be modified, and just using that, what we have been able to find is that the structure of these sort of new lipids guide delivery into different organs, into different tissues. And we have used design of experiments to then formulate. So we have made like 80 or 90 different ionizable lipids, and we formulated with our own, the PEG lipid sterols and others. And what we have been able to do is, using high throughput methods, we have been able to formulate these particles. We have been able to measure their hydrodynamic size, encapsulation efficiency in a very rapid way. So I think where the field is moving, we used to basically test and even formulate the particles one by one. What we have been able to do is now do a high throughput a method for high throughput formulation of these particles, as well as characterization of it. And that's where I feel like the field, is moving beyond what was done sort of previously. And a lot of work has been done. Beautiful work has been done in the barcode by my friend James Dalman, and some work also by Mike Mitchell. And I think that is a very good screening tool to identify what the biodistribution of the materials is. So we have been able to use that to identify certain formulations that, depending on certain parameters, can either go to the lung or the spleen. We have been able to figure out that by changing the lipid structure, we can guide the LNP towards the liver or spleen, or we can guide it towards the lung. Again, this is the same library, but within the library, we have been able to, because there is a rational design to it, we have been able to guide the particle and enable it to deliver, to the liver or to the lung. And recently, we have some more data that some of these materials have their own propensity within the eye as well. So we are very excited to share that in the future with you as well. What my lab is doing currently is designing new materials and understanding the trafficking pathways, using different cargoes for CF, retinal diseases, or SARS-CoV-2. We have methods that we have developed for localized delivery, but I think for systemic delivery, there is a high need to combine a lot of these different parameters together so that we can have some sort of a directed evolution that we can undergo as well. And I think in the next time around, I'll talk about how we think of directed evolution of lipid nanoparticles. Our lab is focused to generate, and train new scientists, pursue high impact science, outreach, and also, open new companies. Again, following the footstep of Pieter and his sort of way of how he brought an ecosystem at UBC. My hope is to bring that ecosystem here in Portland, Oregon. And for that, we have started a Center for Innovative Drug Delivery and Imaging. Again, we have started to get some of the cGMP manufacturing capabilities up and running here. And we have a core team where our focus is not just lipids, but we have polymers, inorganic particles, imaging agents, and are building so that we could do some GMP scale manufacturing of after the discovery phase in the labs to take and translate it further. We have the center. Finally, my thanks to a lot of people within the lab. I showed some work from some of my grad students who now are either faculty or postdocs. Long-term collaborator Renee Martinesi from the Eye Institute. We have more work on polymers and others that hopefully I'll show in the future as well. And funding from the CF Foundation, the NIH, and the NEI. We have also initiated, again, in Pieter's footstep, a company called Enterex. And the vision there is that to bring all these tools of biology, high throughput screening, and chemistry to deliver formulations in a directed and evolution way beyond the hepatic regions and towards different cell types selectively. With that, I'm be happy to answer any questions that you have or any comments. So thank you so much.
There is a question from Gopi Vimuri. I can ask it for him. His question is, how challenging is it for an LNP to reach the epithelium when given via IV?
So I think this is based for the lung experiment. So, and on the IV front, I think we have data where, we have some new materials that we do find have a whole lung sort of distribution. I didn't just show that data. And, there are people who are like, Dan Seeger's beautiful work with SWOT, where they are seeing expression, I think, in multiple cell types, including epithelia as well. It is more challenging than if you just inhale a material, but then you have to go through the mucus environment there as well. So there's no win here. And I do think, again, using high throughput ways, targeting ways as well, we might be able to even boost that tropism. Once we find a particle that is tropic enough to the lung, I think we can boost it further through some of these targeting strategies.
Thank you. Do we have any questions, Marshall, that you would like to ask someone to ask?
Yes, we have a raised hand, Wei Hai Zhang. I'm allowing you to talk. If you unmute, you can pose your question. Please go ahead.
Yeah, thanks very much for this wonderful presentation. I have questions about is IV or is inhalation better than IV for lung targeting?
The answer is, at this point, we don't know. I feel like it depends on what you are targeting. If it's for CF, the answer would be we don't know until we, there are clinical trials happening for inhalation. I think locally it makes sense to do an inhalation first, right? And then if we are able to solve the problem, because there have been even failures in the field where we have tried to make inhalable lipid nanoparticles and they didn't deliver CFTR, for example, in a potent way. So the jury is still out on it, but if I understand, again, through the conversation with, for example, Cystic Fibrosis Foundation, the agenda would be to try both and see if it is possible to do either way. But again, it depends on also what you're trying to solve. Like if it's airway epithelia, it makes more sense to start with the inhalation route first, right? But if it's other diseases that hit maybe alveoli or endothelial cells within the lung, an IV route makes more sense. But again, there is also the challenge, like if you inject anything IV, even though expression might not be happening in the liver, but there might still be the particle there. So there is always an off-target sort of effect. And again, we are trying to develop targeting technologies for a long time within the field, but I do think that with these high throughput ways, we might, be close to success than we were previously. But to answer your question, it depends on what is the disease target that you're going after. For CF, the jury in my mind is open for both. But you start with local delivery first, which is, which many of these companies are doing.
Yeah, thanks very much.
Thank you. There is a question from Marshall, I think from an anonymous attendee. Perhaps we could ask, I don't know if it's easy for them to be enabled to ask their question.
I just can't identify the person to be able to unmute them.
Okay,
so perhaps
then I'll ask the question. I'll ask the question. Okay. So Dr. Sahay, the question is, and I'm not sure I understand the question fully, but the question is, the phage display system seems to work pretty well for local delivery in the eye. Do you think it will work as robust for biopanning screening in systemical delivery?
Yeah, I think that's a great question, and thank you for that. I mean, systemic, it has its own challenges. Immune response becomes a huge issue. But through systemic routes, like a lot of, these antibodies have been found through, integrins have been found for cancer through systemic rounds of biopanning as well. So I think it might, but it might also be that you also already have targeting ligands, but you have to do a lot of modifications in them and do a high throughput way of testing them in a rapid way. If you're able to develop that, that would actually be helpful for the field. But yeah, I think the people have tried systemic delivery and have identified certain antibodies through that route as well.
Right. I see. Thank you. It's clear that maybe I can ask the question. It's clear that the delivery of the nanoparticles into cells and endosomal release finally in the cell that you care about is relatively low. But I didn't get a sense exactly for the difference, or rather, maybe I'll phrase it this way. When you deliver the lipid nanoparticle to the lung through inhalation, could you say how much gets through mucus, how much gets into the cell, and then I guess the cell you care about, and then finally endosomal release? I mean, sort of the stages. Do you know what I mean?
Yeah, I think, look, I cannot say exactly. So here's the challenge of studying mucus in the lung, right? Like mice do not have mucus. Non-human primates don't have mucus. So you have to work with a hamster. So what you do is you take like external mucus and study the transport of the particle. So it's a relatively harder question to answer if you don't do this in a model of existing mucus. And there is one ENAC model for the mice. We have not worked on it yet, but I think there's a huge loss there. I can answer what happens when things are inside the cells, and the escape is, what people have found using, for example, MC3, on Patro formulation, where they injected in the IV and figured out the escape was like less than 1%. For siRNA, papers by Moderna have shown around 10 to 15% escape. So I think there's a heavy loss. I think there's like 80-90% loss before, and then like just 1% actually reaches if we give these nanoparticles into the systemic delivery, forget the mucus, into the cytosolic sort of realm. But it's enough for things like an RNA, which can, these transcripts can make a lot of protein. The siRNA, a little, like thousands of siRNA, can cause a huge knockdown as well. So but it's still a challenge, because I feel like, we end up giving a very high dose still. And if we can improve the escape, we might need 10th of a dose to reach. So I cannot really quantify the mucus loss, but I can tell you the loss, in the endocytic realm, that the escape is less than like around 1%.
Yeah, so you're right. I agree completely. These are complex situations, and you may not need too much once you're within the cell. It depends on what your purpose is. I do notice there's Marshall. Do you see a few more people who are asking questions online here?
Yep, we have 5 written questions and a raised hand. I can, Christian Kastrup has his hand up. I've given you permission to talk. Christian, if you unmute yourself, you should be able to speak. Please go ahead.
Okay, thanks. Hi, Gaurav. It's your friend Christian. Good. Yeah, it's really nice to see how well it's exciting to see how well your eye research is coming along. It's really wonderful. I had a question, of course, about blood. You know me pretty well. I'm sure you know this. This question come from me. Have you seen in the screening that you've done? Have you seen transfection or expression in blood cells or blood cell progenitor cells in the bone marrow? You probably looked for those, I assume.
Yeah, no, I have not really looked into this is a recent quest that we are going for the blood, because what we find is, interestingly, some of these particles and how you formulate polymers versus a lipid, we find their distribution to be weird, for lack of a better word. And my thinking is that we are interacting with some blood cells, and that's why they are carrying into some of those locations. And we're currently working on what happens with certain formulations if we inject, take out the peripheral blood, and see where the transfection is, because I do think that there is an interaction. And in certain cases, I think there might be an interaction, but the RNA might not get expressed, right? Like, for example, the particle might still end up in a macrophage, but the escape from a macrophage, I figure out, is much more harder, even though the uptake is high. So, one thing, again, with things like barcoding and even other distribution methods, is to look for where the distribution of the particle is. and whether then the endocytic escape from there is lower in cause. And what we find is, if I've seen that distribution change based on the cargo as well. So maybe an RNA doesn't express, but a DNA does. So, to answer your question, there is definitely a blood cell interaction. In certain cases, some materials might be able to see expression, and that expression then you end up seeing in the spleen or some other location within the body. But there might be in certain cases that the cargo is not getting expressed, but the particle is still ending up in a certain population of the blood as well. So I think both are, two different parameters that we'll have to look for, but there's definitely an interaction. It's just that we have not really gone after it, and we've recently started to think of that as well.
Okay, cool. Yeah, thanks. And of course, if you ever see transfection of blood cells, as always, let me know, please.
I will. Thank you.
I think we have a few more questions. Perhaps in the interest of time, I'll ask them because I'd like to get their questions asked, but it sometimes takes time. So the question from Shu Jehan is, have you evaluated the pro-inflammatory responses of ocular and inhalable lipid nanoparticles?
Yeah, that's a great question. So we are currently doing that for the eye, and we have also used non-steroidal anti-inflammatory drugs in the eye. Again, not longitudinally, we have just done 48 hours. But in the lung, we did do and have published that. But you have to remember that we have done that. Like for the eye, I'm more confident in saying things because we have reached a non-human primate there. Mice are very sturdy creatures. So to measure an inflammation response there, even though someone asked us, as a reviewer asked us, so we did all those panels and all that study. But to be honest with you, like I can tell you that we did it and we didn't find anything, but it was a mice or a mice model, right? It's not an NHP model for the lung. In the eye, similarly, with MC3 like materials, we do find an inflammatory response like an IBA1 and a CD3 response. As much as we think that eye is not inflammatory, it has inflammatory aspects to it. But with some of our newer lipids, as we pan and go beyond MC3, we find that can be reduced, or the use of like steroids that has also happened in AAV case is helpful as well. So, we're checking for both. And of course, you'll not find much in the like mouse space, but in the non-human primate space, that's where you start looking for those responses.
Dr. Sahay, we have reached the end of our time, but there are several more questions. Do you still have a couple of minutes to answer a few more questions?
Sure.
Okay.
Yep.
The next question, thank you so much. The next question is, have you studied the potential effect of protein corona when injecting the peptide LNP in vivo?
Yeah, I think right now we have done subretinal injection, right? So it's like local delivery. We are working on methods to study the protein corona in the eye space. We have not done systemic injections with them. So, that's why it's more local at this point.
Right. The next question is, and I think you already spoke to this on your seminar, so maybe a quick answer. Does the size of the LNP matter for inhalation into the lung? What's the best size?
Yeah, I mean, look, we think the size sort of, of the LNP as regards to if it's not aggregating is a good aspect, but, particle is also inside like a, fluid, right? Like, because it's a mist in which it's resides. So depending, and there's been a lot of work done there, what the droplet size is, that's where the particle goes in and sits in. So to answer your question, I think it's more on the droplet size rather than the particle size. We just want a stable particle that doesn't just aggregate off, but it's the droplet size that matters when you are doing a nebulization of a material.
Right. The next question is, can you comment on challenges of RNA delivery versus DNA delivery?
Yeah, I think, RNA delivery, less toxic. You go to the cytosol, you see huge expression. DNA delivery is still a challenge. I think we started the field, wanted to go into the DNA realm. We were not as potent, than viral vectors at that time. And beautiful work was done by Pieter and everyone to really bring the field that when the next cargo came in, like SI, we planned for SI and it worked really well. For mRNA, it works really well. But for DNA, I think we still have a long way to go. There is a huge immune response that we see whenever you are working. Like, it's not just one time I've seen it, I've seen with multiple different cargos. So I think it's still a huge challenge. And if someone can resolve that challenge of DNA delivery with a non-viral vector, I think that'll be great, especially if you want long-term expression. Because if mRNA is being delivered as a therapeutic, the challenge has been, how long the mRNA stays. You can engineer the mRNA or the protein, but you still have to do repeated doses. And as much as I love lipid nanoparticles, they have the cationic side to it. How much dose can we really give without having like an inflamed response if given locally, systemically? So yeah, I think right now we have RNA figured out, but DNA is still something of a challenge in my view, because of the immune response that we see.
Thanks. Two more questions. One, for quantifying where LNPs go, have you noticed any differences between the expression of the reporter, i.e. GFP, versus the distribution of the particle itself, i.e. quantified by radio labeling?
Yeah, that's a great question. And I think, this beautiful work by James Dahlman and others have started to, get away from radio labeling, but doing the barcoding stuff. And they do find that the distribution might be different and the expression kinetics is different. They have new work where they are measuring single cell level expression as well. So, definitely there is a difference, but you have to remember, it's a chicken and egg story. If you radio label, is the particle the same particle, or it's different, or even if you put like a code in it. So it's a little harder question. I think the question then becomes, where these plain lipids are. And I think they definitely are going into different organ types. It's just that they have been able to deliver in some of the tissues and cells. And that's the difference you see.
Mm-hmm.
Yeah.
And then the last question that I think we can handle right now. Given the differences between humans and animal models, what recommendations do you have for screening materials to ensure that they will work in humans?
The short answer is working in non-human primates, because that's the closest, and the things that have worked in a non-human primate have worked really well, where LNPs are concerned. Even mice model, if you look at, even with the vaccines or identification of some of these potent materials way back for siRNA delivery, were successful in a mice. They became better in a non-human primate and then better in a human. Now, the challenge again is, like, in a disease condition, things might change. For example, again, in the CF condition, the things would be different. So what is the right model is a very tough question. So this screening parameter depends on what are you going after, and then deciding, like, I cannot give a generic broad sort of answer because it might not reflect. So what target are you going after? Are you going after an eye versus the lung, right? Like, are you going after a certain disease? So what kind of disease? So I think that might help, but the short answer is things have worked well in a mouse model, but if we all had access to NHPs, which are really tough to access, right? Costly, tough to access, that would be a great model to go directly. And it's just really hard to do that, especially from an academic setting, or even company setting. They're very expensive right now.
So I think we need to close the questions now. I want to really thank you, Dr. Sahay, for amazingly great seminar. Especially what I enjoyed about the session was that as you gave explanations of what you're doing, and you gave your description of what you thought was going on. You gave alternative perspectives and alternative points of view for something that might be happening, and that also came out in the question and answer period, as you, I think, gave the audience a chance to see the richness of this field. So I really want to thank you for that. And I invite the attendees to please join me. Please join me in thanking Dr. Sahay for delivering the March 2023 Pieter Cullis Invitational Lecture.